Welcome back guys to my new video on O-level biology. Today we'll be discussing on industrial fermenter. Okay, this is a topic from chapter 21 in your IGCSE book. Alright, so this topic, it has the importance in your exam point of view. Okay, so today we'll be discussing detail on this industrial fermenter. Okay, so there is a picture of industrial fermenter. Okay, so we'll be discussing on each of the point. You can see steam in nutrients. We'll be discussing on that. What is the use of these things? Okay, so first of all you have to know what is a fermenter. Fermenter is a tank. It's a tank. Okay, it's a tank to provide these things. See, temperature, oxygen, carbon dioxide, pH. Alright, so in a fermenter it's a tank. In that tank the temperature, oxygen, carbon dioxide, pH, the nutrients are so these things are provided so that the microorganisms, okay, so that the microorganisms, they can yield their products efficiently. Okay, so industrially, as you know, we grow microorganisms, or we need some products from the microorganisms that can be used. Alright, so today we will be discussing on an important product of penicillin. Industrial fermenter, the biggest advantage of using an industrial fermenter is to produce what? Penicillin. All of you know about penicillin, right? So this is penicillin. Penicillin. The penicillin, so worldwide in the medicine, in the medical industry, okay, penicillin is a important drug in order to create what? Antibiotic. Alright, so the antibiotics, whatever you use, it only comes from a group of chemicals called penicillin, and the bacteria used here, you can call it as Penicillium notatum. The bacteria is Penicillium notatum. So this is the bacteria that produces penicillin. Alright, this is the most important bacteria, you can say, in the history of humankind. Okay, this bacteria, it produces penicillin, and with the help of penicillin we are able to produce what? Antibiotics. Alright, so in industries, these Penicillium notatum, they are what? Nurtured. Okay, we know bacteria, they have a ability to multiply. Alright, so we use that ability of bacteria to produce, like from one fermenter, if you have some Penicillium notatum, and if you provide these conditions, if you provide these conditions in the optimum level, okay, the Penicillium notatum will yield penicillin. Okay, then we extract that penicillin and use that penicillin in the pharmaceutical industries to produce what? Antibiotics. Alright, so this is the whole scenario for using an industrial fermenter. Okay, the industrial fermenter not only used for producing penicillin, but even for producing more bacteria, genetically modified bacteria. In next videos I'll be discussing on that. Okay, even the bacteria, all of us, we have wrong concept that bacteria are harmful, they cause disease. Yes, they cause disease, but they are very helpful in another way. In which way? If we genetically modify them, they can be used in a useful manner. Okay, in the next chapter we have those aspects. Okay, in chapter 22 I'll be discussing on that. Alright, so for the time being, this tutorial is for industrial fermenter. So what is an industrial fermenter? So now you have an idea. It's a tank, alright, in which these conditions are maintained so that the bacteria, they can yield or produce the what? They can produce the products efficiently. Okay, now let's look in a detailed manner what is a industrial fermenter is. Okay, in industrial fermenter they have a steam inlet. So there is a steam inlet. Okay, so what is this steam inlet used for? Before starting, let me clear this. In industrial fermenter, we perform a batch process. Batch process. What do you mean by batch? Like a batch of thousands of Penicillium notatum is inserted into the industrial fermenter. Then they are nurtured, okay, and they produce what penicillin. After their production, they are removed from the fermenter. Okay, they are removed. The whole batch is removed. Then new batch has to be fed inside the fermenter. Okay, so this steam inlet, the steam in, or you can call it a steam inlet, is used to pass hot steam through the fermenter so that any microorganism remaining inside the fermenter would die. Okay, the steam inlet is used to wash the fermenter, you can call it. Okay, so this is the steam inlet. What is the use of steam inlet? Used to clean the fermenter. Okay, the fermenter is cleaned with the help of hot steam. Alright? Now, what is this nutrient column? This nutrient one. Let me write it with blue. Okay. What is this nutrient inlet? As I said, this Penicillium notatum is nurtured in industry for what? In order to get this product, penicillin. Okay. Now, this Penicillium notatum, it needs nutrients, or you can call food for Penicillium notatum. The bacteria, the Penicillium notatum, it needs food. Alright. This nutrient is the food for Penicillium notatum. This one is the nutrients you can call. This is a mold. Okay. It contains food for what? Penicillium notatum. So the food is inserted into the fermenter through this pipe, alright, or through this inlet. Clear? So this is your mold of what? Penicillium notatum. Here Penicillium notatum is present and the nutrients, alright, on which the Penicillium notatum will feed and produce what? Penicillin in an efficient way. Understood? Now this is done. This is done. Now let's go for this. What is this? Water jacket. So this is your cold water jacket. The bluish part on the both side of the tank. It's the water jacket. It basically contains what? Cold water. Now the water jacket is used to cool the fermenter, because fermenter, it produces a lot of heat inside the tank. Why? Because these Penicillium notatum, they respire aerobically, or even you can say anaerobically sometimes, alright. So because of respiration, or you can say these Penicillium notatum, because of they respire inside the fermenter, they produce a hell lot of what? Heat inside them, because there are millions of what? Penicillium notatum inside the fermenter. Clear? So as a result, you need this cooling water jacket. Okay, there are water jacket so that the fermenter don't get heated up and burst. Okay, because as you know, if the heat gets added up, they can burst the fermenter. Alright, clear? So this is the use of water jacket. Now let's go for cooling water inlet. Now you will be saying, why do you need a cooling water inlet? See, this water jacket, I said it's a jacket that contains cold water. Now bring on physics here. The cold water will get heated up because it's absorbing the heat from the fermenter. Alright, so this water doesn't stay cold for its whole time, right? For the whole time of the process, this cold water doesn't stay cold for the whole process. It gets heated up. Now you have to remove that heated water, okay, through which this is that. So from the jacket, the water is removed. Here hot water is removed. You can say hot water removed. Okay, so the water from the water jacket, that is cold water, that gets heated up and gets removed from this pipe. Okay, and new cold water is needed, and that's the cooling water inlet. Okay, feed again through this inlet, the cold waters are run through the fermenter. Okay, now let's go for air in. What is this air in? This air in is important. First of all, this is the general fermenter used in the industry. Okay, there are many organisms, microorganisms, they need oxygen for what? For respiration. In this case, you might say that Penicillium notatum doesn't need oxygen, so doesn't need oxygen for respiration, so they respire anaerobically. Okay, so in the case of Penicillium notatum fermenter, the fermenters that are used to nurture Penicillium notatum, you don't need this air inlet. You don't need this air inlet. But in the case of other organisms, as I mentioned before, that industrial fermenter is used for producing more microorganisms like bacteria or any other kind of bacteria or fungus. They might need oxygen for what? Respiration. Okay, so in that case, the oxygen inlet, you can say, this is oxygen inlet. Alright, the fresh oxygen is inserted into the fermenter through this pipe. Okay, now you have to remember something. For O-level point of view, they might ask you some questions that this oxygen that are inserted into the fermenter, okay, they are filtered. These are filtered oxygen. These are your filtered oxygen. Why do you need to filter the air? You filter the air so that any foreign or any microorganism mixed with the air cannot enter the fermenter. Okay, as a result, the air going inside the fermenter is filtered so that no microorganisms from outside enter the fermenter. Okay, now you will be asking, what will happen if these outside microorganisms they enter the fermenter? If any foreign organism enters the fermenter, it will compete with the Penicillium notatum or any other bacteria which you are nurturing. Okay, it will compete for nutrition. Understood? As a result, there will be two problems. What are the problems? First, bacteria which you are nurturing, okay, it will hamper their production. It will hamper their production. It will hamper their growth, you can say. Secondly, the products will be contaminated with the waste from the foreign microorganisms. Okay, the product, what do you mean by product? Product, I'm saying this fermenter is used to produce penicillin. Understood? So if foreign microorganisms they enter the fermenter, okay, they produce a hell lot of waste. Okay, so the penicillin will be contaminated. The product will be contaminated, you can say. Product contaminated. Okay, the products might get contaminated, so as a result the product will not be pure. Okay, so for that reason we filter the air so that no microorganism could enter the fermenter. Okay, now understood. So water cooling over. Now what is the use of this steering paddle? Steering paddle, what is the use of that? Let's go for reddish. Okay, the steering paddle is very important. Okay, steering paddles, they mix the nutrients evenly. Okay, they're used for steering the nutrients inside the fermenter. By mixing the nutrient, what happens? The nutrients are evenly spread throughout the fermenter. Okay, when this steering paddle, they evenly distributes what is the use? They evenly distribute the heat. You can say heat. They evenly distribute the nutrients. Okay, they evenly distribute the nutrient, and they help to keep the temperature of the fermenter to a normal level. Okay, understood? So that's why these steering paddles are used. Okay, so they ensure so the temperature throughout the fermenter is even. Understood? This is the use of steering paddle. Alright? Now what is the use of this stainless steel? Now, what is the use of this stainless steel? You have to understand these Penicillium notatum or any other bacteria, after they respire, as you know, respiration comes with some waste. Okay? When their cell respire, they produce a hell lot of what? Waste in them. Okay? So these wastes are acidic. Okay? The waste from the microorganisms are acidic. So in order for the fermenter doesn't corrosion. Here, stainless steel is used so that the steel of the fermenter doesn't corrode. Alright, and the final one is the product outlet. As you know the name, it's the outlet for the product. When the penicillin is produced, they remove through this pipe. Clear? Okay. Yeah, that's all. For O-level point of view, that's why I have mentioned so many points. Okay, this kind of questions they are asking. They ask like, why is the steering paddle useful? Why do you have to maintain the pH of the fermenter? I've seen some questions. First, remember something. If the pH level is not maintained, the bacteria would get denatured and they will not work. That means the Penicillium notatum won't produce penicillin if they get denatured. As a result, you have to maintain the pH of the fermenter. So if you like my video, do not forget to subscribe to my channel. Okay? Yeah, because I'm making some useful topics, right? It's very useful for you.